Okay, I want to thank everybody for attending the Symbiota Working Group webinar today. Can hear? Okay, so everyone can hear except for Jenny. So you may want to fiddle with your microphone there, your speakers, Jenny. So today we'll talk about a new GenBank submission tool that's recently been developed by Phil Anders. He's my developer here at the University of Illinois. And it's something where we found an issue and we were hoping to be able to solve it with this new tool. So I just wanted to introduce everyone to this today. So I'm just going to start out with a brief introduction. Maybe some people aren't aware of the vastness of Symbiota. There's at least 767 collections with more being added, it seems, every week. These include over 37 million records. There's about 40 portals right now, 13 of which are TCN portals in the Thematic Collections Network. So you can see most of these are either taxon-based or they are either regionally based type portals. But we have a large amount of metadata, obviously, right, within Symbiota right now. So we want to be able to take advantage of that and use that to our best interest. So just as a brief introduction, I talked about this, I think last year or maybe the year before, linking data between Symbiota portals and GenBank. And I just wanted to make everybody aware that we do have this feature. It's available in all the portals right now. If you go to your occurrence editor, occurrence record editor, and click on the linked resources tab right there. So I can try to make use of my pointer. So so the link resources tab right there, you'll see that you're able, excuse me, you're able to add genetic resources by clicking on the pencil, filling it out, and filling out the information. So I'm not going to go over all that right now. Again, you can refer back to the other Symbiota webinars, the one that I gave on linking genetic data, for more information on that. But when you do go ahead and link a record, first I just wanted to point out that in the future, if you do decide to upload this plugin tool to your Symbiota portal, this is where you're going to end up finding this tool at. So there'll be a button that says Submit Sequence to GenBank. It's not there yet in any of the portals, but it is coming. So this is just a demo today on how this thing works, but it will definitely be in the linked resources tab that you can click on it and then go and use this tool. So I understand some people are having trouble hearing, and that's most likely, it seems like it must be on your end because everyone else seems to be hearing me loud and clear. So not sure what's going on with that, but I'll just keep going because this is recorded. You can always go back and listen to it more. So so anyway, that's all I'm showing is where this button will occur down the road. And if you click on this genetic link here, so right here, this URL, if you go ahead and click on that for anything that has genetic data, it's going to take you to the GenBank record, right? So here's your GenBank record, and then what you're most likely going to see over here is the Mycology Collections portal in the list of link outs, okay? So, for example, Mycology Collections portal is a dedicated link out, and we can link back and forth between GenBank records and the Symbiota portal, the microportal. So if you click this link, then what that's going to do is link you back to the public display interface. And we didn't link it back to the occurrence editor for obvious reasons. You need to be logged in order to use the occurrence editor or see the occurrence editor. But this would link you back directly to the public interface right here, and where you would be able to see all the specimen metadata. And then if you click on the genetic data tab, obviously it shows you all the genetic data that is very similar to what you just ended up seeing in the occurrence editor. It's basically pulls from the same table. Okay. So that's all I wanted to remind people of, is there is ways right now to link back and forth between Symbiota portals and GenBank records. It's not automated. We're hoping to improve that in the future, though. So to begin my webinar today, this is what I wanted to pretty much show is the problem. First state the problem and then later state what we believe is the solution. And as many of you probably already know, source modifiers are seldom populated in GenBank records. And by source modifiers, what I'm talking about is basically the specimen metadata fields that GenBank refers to as source modifiers. And these are the most relevant to us: the specimen voucher, the country, the isolation source, the host, collected by, collection date, identified by, latitude, longitude, and altitude. And for those of you that are Darwin Core savvy, the first thing that you'll recognize is these are not all one-to-one relationships to Darwin Core terms, right? Especially country. Some of them, most of them are one-to-one relationships. So a specimen voucher can be tied directly to catalog number. Isolation source is going to be pulling directly from substrate. Only the mycoportal, the fungal people, you actually used host. And we can discuss that later on. Basically, we decided to put another field into our Symbiota portal called host. And that's what it pulls from to serve GenBank records. Collected by, collection date, identified by, lat, long, and altitude. These are all pretty self-explanatory in how they're one-to-one relationships and how they match up to Darwin Core. What we're doing as far as country goes, GenBank puts all of the locality data under one single source modifier called country. So that's basically what we did, was concatenate the country field, the state, province, the county, the municipality, and then the locality field are all concatenated together into country. And that's on GenBank's end, how we had to do it. So this is the problem. So this is one example here. This is a fungal record that is pretty good about populating most of the source modifiers. So what we can see is isolation source, or the substrate, which happens to be a stem, is populated. The host, which is some plant species. Specimen vouchers populated. They did list the country. They list lat and long, and altitude, collected by, collection date, and collected by. So I think the only one they're missing here is just identified by. So that's pretty good. It's a nice GenBank record that's fairly heavily populated with good specimen metadata. And then you have this example by some bozo back in 2004, probably some grad student that didn't know any better when he was uploading his GenBank records, and only populated one single source, and that was specimen voucher, right? So obviously A.N. Miller could have done a better job at populating the metadata associated with his GenBank record, but he's not the only bozo out there, as we'll soon see. So what I did was, just to test to see how much metadata was actually in GenBank, is I was using this in-tray search string that got rid of whole genome data and some of the other GenBank databases. But I was mainly just searching in the main GenBank record where most Sanger sequencing sequences end up. And for a fungal data set, we see the mycologists are pretty good at populating the specimen voucher source modifier, right? So 82% of the time, you can look at a GenBank record and link it back to at least some number that hopefully links back to a specimen voucher that occurs in some fungarium or mycology collection around the world. We're not so good on country, only about half of the time that's populated, and then it kind of goes downhill from there, right? So not much specimen metadata. So the mycologists aren't doing too bad, but not very well. The entomologists are even worse when it comes to specimen voucher. So what this shows is that only less than a third of the time, if you're looking for the actual specimen where the sequence was generated from, less than a third of the time you're actually going to be able to find that for arthropod data sets. Entomologists seem to like country and collection date. They do a good job of populating that, but everything else not so much. Okay, and for you botanists out there, unfortunately you're equally bad. About a third of the time you actually put any data at all under specimen voucher. to link this data back, the sequence data back to some specimen voucher. And then, of course, everything else is populated pretty low. And last but not least, the vertebrate people out there. Same kind of thing. Not a lot of data out there, metadata associated with your GenBank sequence records. So obviously, this is a problem. We would like to have the metadata populated in GenBank records and not have it lying in some other unlinked resource. Excuse me. So the solution is obvious when you think about it. You just pull the metadata directly from a collection management system such as Symbiota, right? And then submit that data to GenBank. If this data is already out there in some collection management system and you spent the time typing it in or transcribing it or whatever to get it into some sort of data portal, why would you want to go ahead and retype it all over again? It doesn't make a lot of sense to do that again when the data is already sitting there and we can just pull that data automatically. So that was the idea behind this tool and what we ended up developing. So here's kind of a schematics. Sorry about the center arrows, they never want to line up right. But basically this shows a schematics of what I'm going to show a little bit later in my webinar when I show the tool live. And what this Symbiota submission tool is going to do is it's going to automatically pull your user profile information. As long as you're logged in to a Symbiota portal, it just pulls that automatically. And then it's going to take your specimen metadata, and whatever is populated in your Symbiota portal in your occurrence editor, it's going to pull that data. So obviously if that data is wrong or not populated, it's going to pull incorrect data. So you're going to want to look at your data and just verify it, confirm it one last time before you actually submit a GenBank record with that specimen metadata. And then you basically cut and paste in your sequence. You download the file and you email it off to GenBank, just like you would a sequence file. So it's a pretty simple tool that we developed that doesn't have a lot of roadblocks in the way for making it work. So for those of you that may not be familiar with different ways to submit your data to GenBank, there's 4 primary methods. One is Bankit, and I don't really know much about Bankit. I don't really know anyone that's ever used Bankit. I checked it out last week for the first time, and it looks like a precursor to the GenBank online submission portal. It's pretty easy and straightforward and simple to use, but doesn't seem to be widely popularly used, at least in my community. And then third down is this online submission portal. And like our tool, it's only used for ribosomal data right now. So we're talking small subunit, large subunit, and ITS. You cannot use it for submitting sequences of protein coding genes, and the same thing for our tool at this time. And then finally you have Sequin, which is currently being phased out by GenBank. So Sequin's the standalone program that you download onto your desktop, but it's not going to be available in the future because it's being phased out, and GenBank is moving more towards the online submission portal. So what we ended up using was GenBank's own code, actually, this second one down, called table to ASN. And basically what we're doing is just submitting data to table to ASN code and letting that code generate the Sequin file. So this was a lot easier than trying to create our own code, and it made working with GenBank a whole lot easier because we're using their tools that they already developed. So for those of you maybe not familiar with the online submission portal, if you haven't checked it out yet, it is pretty nice. It's very slick. GenBank seemed to do a pretty good job with this, and it's basically just a series of different tabs up here. So you have like a submission type tab that you click a lot of things, and then submitter, that's where your user information, analogous to user information, sequencing technology with a drop-down list. This is where you cut and paste your sequences, and then your source modifiers tab where you populate that. You can put in some references if it's published, if your sequence is published or going to be published with citation authors, And then you can kind of click on the overview and see some of your files, but that's somewhat limited. So if we look at the source modifier tab, though, you'll see this is just a screenshot from what it actually looks like. And you would type in your organism name and then your specimen voucher, your barcode or catalog number, whatever that would be. And then you have other source modifiers. But the problem is you have to go on and select each of these one at a time and then type that in manually. And this is just introducing more user error, right? So what would be the point? You've already typed this in once into your Symbiota portal, or someone has. So the whole idea is just to pull this data automatically from Symbiota instead of using the online GenBank submission portal and typing it all over again, where you can introduce more errors. So that was kind of the idea behind our tool. I just wanted to point that out to kind of make it clear what you would have to end up doing in GenBank to do that. I'm also going to show you later in my live demo that our tool will also generate something called a TSV file, a tab separated value file. And you don't see it on here because I'm only submitting a single sequence. If you submitted multiple sequences on this page, you would see a button that says upload TSV file. And what that would do is allow you to upload a TSV file that had all of the different source modifiers in it for each of your different sequences, right? So our program is generating that, but only for one sequence at a time. And in talking with GenBank, they realized that maybe they should also offer that option as well. Even if you submit a single sequence, you still should be able to upload a TSV file with all of that source modifier data populated. So their developers are actually working on it, and I was surprised how congenial GenBank actually was to meeting the user community needs and working on this. So hopefully in the near future there'll be a button here where if you don't want to use our tool to submit the sequence, you can still use our tool to generate the TSV file with all the source modifiers for your specimen, and then you can upload that file automatically so you don't have to go through and type it in all over again. Okay? And I'm happy to answer any questions about that after the webinar if that's not clear. Okay, so just some plugs for this tool. Why you might want to use this tool versus GenBank's online submission tool. It's fast and easy generation of an SQN file, and I'll show you that in a minute. Again, as I've been talking about, there's automatic population of the source modifier metadata that's already been curated and validated data, right? So in Symbiota, there is some validation. You can't just type anything into every field in Symbiota. So this data is validated and curated. Many of us are checking each other's data and we're finding errors, and we're going back and fixing those in the Symbiota portals. So this is actually a very good data set in the Symbiota portals to pull from. We also developed a structured comment that is available for any of the 40 portals. It's going to be registered hopefully next week in GenBank. We've already submitted all the data. They said it looks fine, and they're just working on the developers to implement this structured comment. And I'll show you what that looks like later when I give the online demonstration of this tool. But basically, this structured comment is only going to be available if you go through and use this Symbiota GenBank submission tool, not the online tool. And something that our tool allows you to do that GenBank does not is to preview your GenBank flat file before submission. So if any of you have submitted a sequin file, you know that you can take a look and see what the flat file kind of looks like, what the end product's going to look like in sequin. But for some reason, they don't give you that ability in the GenBank online submission tool, but our tool does. So those are just some of the reasons why I think you might be interested in using our tool. So, all right, I'm going to now switch to an actual live demo, so please be a little patient with me. I'll stop sharing here, and I'm going to share my screen, which means I'm not going to be able to see the chat box very well, if at all. And that means I'll probably be more interested in taking questions towards the end of my webinar. If you could maybe You can go, feel free to go ahead and chat, type them in the chat box now, but I'm probably not going to be able to see the chat very well, and I probably won't want to follow along with it, although I will try my best here. Okay, so obviously this is the portal I work in, the Mycology Collections portal. So it's the one that I chose to use to demo this model. It's actually up and working right now, and only 4 users can actually use it right now. There's a filter placed on it, so not anyone can go on and just start submitting GenBank sequences through the Myco portal right now. But I'm demoing this through the public search interface because that's how we originally designed it. But then we started thinking, why not take advantage of Symbiota's user permissions? So like I showed before, the Submit Sequence to GenBank button is actually going to be under the Linked Resources tab in the Occurrence Editor file. So if you have permissions for a collection, you're going to have permissions to submit a sequence for any of the metadata for the specimens in that collection, right? And I'll show you why that's important in a second. So if we go to Search Collections, I just want to search for a collection here at the University of Illinois. Okay. Go back up, hit search. I'm going to type in my catalog number. And again, you can do it this way to search for your specimen that you want to submit a sequence for, or in the future you'll be able to use the occurrence editor. Okay. So we do a quick search. Here's the one that pops up. This is the one I want to submit my sequence data for. Like I said, you would hit Occurrence Editor and go to that Link Resources tab to find the button in the future, but for right now, we just threw it under the Genetics Data tab, and it says Submit Sequence to GenBank. So by putting it here, you Symbiota savvy people will realize that anyone would pretty much be able to submit a sequence for anything in that portal if we put it on the public interface. So that's why we're going to remove it from here in the public interface and put it a little more behind the scenes in the occurrence editor record. Okay, so you basically click on that, and this is a plug-in module that runs outside of Symbiota. There's actually only, I believe, 5 lines of code that you have to put into the Symbiota portal to get this tool to work. So either Ed or Ben can help you with that if you would like to take advantage of this tool. So the first thing you notice is the username is already populated, and that's because I'm logged in, right? So this has already automatically pulled all of your user information. It also pulls the OCC ID, or the record ID for that specimen. And what this has done is already pulled in all the specimen metadata as well for that particular record. If you wanted to go look at the metadata, you could have done that through the occurrence editor and just verified that everything looks good and things are spelled correctly. The next thing you'll want to do working down is to submit your sequence. So I kind of have my sequence all handy and ready to go here. It's just a simple FASTA formatted sequence. We even tell you how to submit or what a FASTA formatted sequence looks like. It has to have that greater than sign and then genus with an underscore and species and a hard return, and then you type in your sequence, for those of you that weren't aware with that. So I just copy in my sequence here, paste it into the box. There it is. It looks fine. And I get lazy and say, I just want to submit this sequence. I don't want to fill anything else out. And it's going to come up with an error message. It's a very simple error message, but it's basically, if you can't read that, I apologize. What it says is, please fill in the form with your name, ID, sequence definition, and sequencing technology are required. So those are the 5 minimum things that you're going to have to submit with your sequence in order to use this tool, right? So I go back, and I want to tell it what the sequence is, actually. You can look at this, you can click on, or not click, just hover over this little question mark, and it actually tells you what we're talking about here. The ITS, the LSU, SSU, or if you have ITS 1 or 2 independently. Those are your options of what you can choose. So this is a large subunit sequence, so that's what I'm going to submit. If you're wondering about any of these definitions on here, these were all approved by GenBank. They had us change it a little bit because we had 28S and 18S in there. That's not what they wanted. So this conforms to what GenBank indexers are looking for and want. So it makes all of our life easier. There's a drop-down list for the different ways of how you might have generated that sequence. This was a Sanger deoxy sequencing, so Sanger sequencing method. And then you have the option of whether this is published or not. And it's just a little slide bar you just kind of click. And that's where you can type in the publication, right? So this one might just be, I don't know, fungi of Colorado or whatever you want to type in for your publication, right? Which you might want to spell correctly. And then adding additional authors. It's important to remember in any GenBank record that there are contact authors and there's citation authors. The contact author is the one above who's logged in is automatically the contact author. That's the username. The citation authors refer to the authors of the publication. And we're not going to assume that your graduate student or your volunteer that might be submitting your GenBank records for you should necessarily be an author on the publication. All right, so we're not automatically populating that. So if you want to be an author on the publication as well, you just simply type in your first, middle, and initials and say add author, right? And then you can add multiple authors if you want, right? So I published this with Jack B. Nimble and Jack B. Quick, a couple of my buddies that helped me out with this, right? So these are your publication authors down here, and there's little X's by there. If Jack B. Nimble upset you last week and you don't want him to be an author anymore, you can just click that and get rid of him, okay? So that's how you add additional authors and the title of the publication. If you want to embargo this for up to a year, it's another simple slide switch. We're not giving you the option to embargo it for 1 week or 3 weeks or a month or whatever. It's basically taking the data of your computer and adding 1 year to it. So you can either embargo it or not, but you're only going to have the option to embargo it for up to a year, and that's pretty much what GenBank allows as well. Okay, so then when you're happy with your submission, you click the Add button. You'll see the collected items are displayed here. If you're not happy with it, you can click this little X right here and get rid of it and start all over again. But if you are happy with it, you click Generate File, and it generates all the files. And then you just want to scroll down a little bit more to this Open the File Manager, okay? And this is where you're going to retrieve your sequence. So this is a dedicated file manager that kind of works behind the scenes to collect all the data. And it just takes a little bit getting used to, but you can see it's like any other very simple file manager. So you would click on your file right here, and this is basically the only place you really need to go to get all the data that you need. And the only data you really need is this sequence file right here. This is what you're going to download and end up sending to GenBank. So you would just kind of run over here to the little download button. If you hover on it, it says download, and you would end up downloading that sequence file and sending it to GenBank. The other options is you can look at this TSV file I was talking about earlier, and basically that is your table separated value file. You can download that, and like I said, in the future, if you wanted to just download that and use GenBank's online submission tool, you could upload this file for that sequence, and you wouldn't have to type in all your source modifiers, okay? So you can see that we pulled the organism name, the altitude, collected by, collection date, country, the host, isolation source identified by, lat long, and specimen voucher, okay? So maybe not, we haven't populated everything in our Symbiota portal, but we pulled everything that we were able to pull. You can click the back button. If you wanted to look at your, excuse me, look at your sequence, you can. Again, here's the name. It's now populated with what that gene is, and then your sequence. You can look at the sequence file. For those of you really savvy, you can scroll through that and look at that. It's basically the same thing that you would end up seeing in a sequence file. But that's the entire file that the table to ASN code has produced. But this is the nice thing that I like to look at, this GBF file. And the GBF file is basically your flat file, okay? So I apologize now if this looks small. I try to blow it up a little bit. There we go. Maybe this will help a little bit to be able to see it. But basically, this is what it's going to end up looking like when you submit the thing to GenBank and they end up publishing it online. This is your sequence record. So first you have your citation authors up here, me and Jack B. Quick right here, along with the title of our publication. The citation author is below that. And then you have your structured comment, which starts with Symbiota specimen reference, start, and then Symbiota specimen reference end. And that's the structured comment that GenBank is now registering for us. And it basically pulls a short URL to your portal, whether that's the Lichen portal, Bryophyte portal, Myco portal, whatever. And then it's going to pull the GUID, so the occurrence record or the GUID for that record if one exists. Of course, many of us are snapshot, or a lot of the collections are snapshot, and they may not have generated a GUID or populated the GUID in their Symbiota portal. So that's up to you whether you want to do that or not before you submit a GenBank record. It will end up pulling the record ID because every record ID in Symbiota is populated when you upload your data. Some of the record IDs might be the same as the occurrence IDs, and that's okay. In our case, it is. I just had Symbiota populate our GUIDs for us instead of getting them from an outside source. You also have institution code, so that's going to be your abbreviation of whatever your collection is. We do not have a collection code. For example, LILL could have a collection code for fungi and plants that would be F or P. If we did have that typed into our collection data on the collection metadata, if we had an F for our collection code, it would show up here. So under institute code, it would say collection code with an F. And then we pull the catalog number and other catalog numbers. So we're just trying to pull as much data as we possibly can. And then below that, you're going to see all this beautifully populated source modifier data, right? So you're going to end up seeing the isolation source, the host, specimen voucher, the country as it's concatenated together with all the Darwin core fields, the lat and the long, the altitude, the collection date, collected by, and identified by. So all of your metadata for that specimen record is now populated and ready to go to GenBank. Okay, so that's pretty much what I wanted to talk about today. I'm going to leave that up, but I'm going to stop sharing here. Let's go back and see. Let's see. It says share. Stop sharing. Okay. So that's pretty much what I wanted to present today to everybody, and I guess I'll go ahead and just open it up for any questions. I do appreciate the wonderful turnout today. It looks like we got about almost 40 participants today. So Patrick would like to know, can other sequence types such as TEF be added later to the drop-down list? So that's a great question. Our goal for this project, which took about 6 months to develop, was to get it to the state of ribosomal genes that we could submit through this tool. And we haven't taken it to the next step further, which would be protein coding genes, and the ability to submit those through this tool. And mainly the reason is it just becomes much more complicated with taking your nucleotide sequence and translating it to amino acids and a lot of other things. So yes, Pat, it certainly could be done to include protein coding genes in this tool, but we don't have any plans right now at the moment to go ahead and do that. This is very similar almost identical to the GenBank online submission tool, in that you can only submit the ribosomal RNA genes right now, just ITS, LSU, and SSU. So, but I'm sure we'd be more than happy if there's some developer out there that wants to take this on to the next step. We'd be more than happy to work with them on try to make that happen, because certainly that would be a very useful thing to add on to this as well. So Scott wants to know, are there plans to concatenate multiple sequences into one TSV file so that they could be submitted via the GenBank portal? Yeah, that seems like a simple thing to do, and we probably could end up doing that. You could concatenate them in bulk so that if you had multiple TSV files, you could actually list those one after the other. So you could either do this in our tool, or you could just generate those TSV files and open them up in Excel and just cut and paste those in either way. It seems like for right now it might be a little easier just to go and cut and paste them into Excel. It might be easier to move them around that way and then just resave it as a TSV file. We could concatenate it, but you would have to make sure that you upload your sequences in GenBank the exact same way that you concatenate the TSV files, right? So the main problem with that, Scott, the thing I don't like about that is that you're using our tool to generate the TSV files, then you go and use the GenBank online submission portal to upload that TSV file, and you're working in two different platforms now. You would just have to make sure and keep straight that you didn't upload metadata for the wrong sequence, right? You would have to make sure and keep those all in order. That's one thing you might want to look out for. But you could certainly just do that in Excel right now, or we might think about adding that to our tool as well. All right, so Deb Lewis, did I understand correctly that the genetic data tab is using table ASN for linking? No, not exactly. So the table to ASN is only used to generate the code to submit the sequence to GenBank. The genetic data tab, though, you are correct, and that's how we end up linking to and from GenBank. It's a little more complicated than that, but basically what happens is GenBank sends us basically a six-digit number, which is a GenBank accession number, and in the end we end up through an Excel spreadsheet that I send them periodically to the link-out team. I email it to them periodically. I'm basically sending them a bunch of data, but the only thing that changes is a six-digit OCC ID number. So we're basically exchanging six-digit numbers back and forth, and we're talking with GenBank now on how to automate that a little easier so the linking could be done automatically. But in the end, that's what it pretty much boils down to, is linking between a Symbiota portal and between GenBank comes down to basically just a couple six-digit numbers. So the machine should be able to do that automatically instead of having people do it manually. So we want to see, now the tool seems to be only for RNA and ITS, but for protein you also need to annotate. Yeah, that's very true for annotating the protein coding genes. So Hojun elegantly pointed out some of my hesitation about taking the next step and trying to include protein coding genes as well. It's more complicated than just a few clicks. So Jason would like to know, does the process of generating a GenBank submission file also create a record or link in Symbiota? Yeah, so ideally that would be the whole idea. And that's one of the next things we need to try to work on. We've been thinking about that and how to do that. But you would be able to fill out all the fields in Symbiota, but you wouldn't be able to fill out, well, you'd only be able to fill out 2 of the 4 fields in Symbiota. And I can quickly just demonstrate that since I have this up and running here anyway. So Jason, what we're talking about, if I just go back to what we populate, so if we go here under Link Resources and you click the little pencil tab, you basically have 4 fields to fill out. And the first field is the name of the source, which is almost always GenBank. unless you're submitted some sort of sequence through Bold or EMBL or some other data source. It's almost always going to be GenBank. The next field is identifier, and that's what you need from GenBank. So you can't really create your record or link in Symbiota until you get that GenBank record, which takes 24 to 48 hours, right? You can fill out the locus, just like you can fill out GenBank. You can fill out that field, but you also can't fill out URL. So you can only fill out 2 of the 4 fields, or 50% of the fields here, until you get this, until you end up getting this GenBank record back again, or GenBank accession number back again. So, and that's, like I said, 6 digits plus 2 letters. I guess it's 8 digits really, an 8-digit number, and that's sort of the bottleneck for us doing any sort of automation. Is if we submitted a field and got an automatic GenBank number back automatically, we could create these automatic links, but that's the holdup. So that's what we're trying to work with GenBank to figure out. And then Linda wants to know, is there any validation that the sequence is truly the RNA that is proposed in the definition line? No, not validation on our end. We're just trusting the Symbiota users to cut and paste the correct sequence up. Now what will happen when it goes to GenBank is they'll send you back a GenBank accession number almost immediately, but it can take several weeks or months, even if you did not embargo the data, it can take a while for them to validate and curate your data. So it would end up being caught on their end. I see we have a GenBank representative here that can correct me if I'm wrong, but I'm pretty sure the indexers would look and see that the product says ITS data, and then they would look at the sequence and realize, oh, this is actually a large subunit. So this is incorrect. So I guess we're expecting, we don't really have any tools inside this tool to look to see what the sequence is and whether it correlates with the definition line. So Hojun said, often when I submit GenBank, they come back to me with some errors about my data, how sequences are annotated, missing data. So when the submission is automated, how is this feedback loop going to work? Well, the submission isn't automated now, so I can't really answer that question. What you're basically doing is generating an SQN file, just like you would do from Sequin, and you're going to email that to jb-sub for GenBank submission, right? Just like you always have submitted your sequences if you use Sequin. So you're basically going to receive an email from GenBank with the errors just like you currently have. None of that has actually changed at all. That's all still the same. But yeah, very, very good question. And we have tested this with GenBank with a few test references, and we also had a teleconference with GenBank with the indexers, asking them, you know, what do you want us to change? How can we improve this? What's right and what's wrong? And they seem quite satisfied now with the changes that we made, and they seem to like this tool. They were quite impressed with the quality of the data, which, like I tried to explain to them, this is all curated and validated data, so it should be pretty high quality data. And because we're pulling automatically from the source data, it should definitely go in the right place, right? The source modifier should be populated correctly. Some of the issues that GenBank has is they said you wouldn't believe what people put in an identified by field. Well, you can't do that anymore unless you manually go in and change your Sequin file. It's going to pull identified by correctly from Symbiota, assuming that you put in the correct metadata in Symbiota to begin with. So, okay, are you posting accessions that are embargoed? Let's see, posting accessions that are embargoed. Oh, you mean keeping track of that somehow? No, not really. We just leave that up to the individual Symbiota user. But I'm sure there's probably a way, since we're collecting this data, there could be a way, I guess, of keeping track of what users submit what sequences with with embargo data in it, I suppose. But no, we haven't thought about that. So not to miss Hojan, GenBank also needs taxonomy. Can this tool auto-populate this information? Yeah, yeah. GenBank has their own set of taxonomy, so that's not up to us. That's a GenBank's end, because they use their own taxonomic thesauri. So that has nothing to do with us. Sorry about that. Lori says, can I ask what identified by means? In GenBank world, it means the person who has actually identified that species, so it should be very, very limited use. Any of the astute Symbiota users on here are more than happy to correct me if I'm wrong, but that's exactly how we're supposed to be using it. At least that's how I use it. Identified by means the person who actually identified that species. So Susan says, if we do, we will have to release it, which is why I asked. Posting sessions that are embargoed. Oh, I see what you mean. Okay, sorry, Susan. Now I understand the question. So if someone goes and embargoes it for 1 year, but then they decide to go into the linked resources tab in Symbiota and post this online, then that would make a lot of sense. But I understand what you're saying now. Is you could have something embargoed that actually has a URL in another portal or another online resource, such as a Symbiota portal. I would guess people wouldn't want to do that, but I wouldn't have any control over the users doing that. I used to embargo my data, but now I don't even bother with it anymore. I just go and release it immediately. So unless it's really sensitive data, I guess, that you're trying to publish, I don't see much reason to embargo in data anyway. It just makes life a little more cumbersome for everyone. So Hojan, I might have missed it. Is there a batch upload mode, like 100 sequences representing different species? Nope. So you didn't miss anything. This is actually one at a time. Instead of high throughput, this is sort of low throughput right now. And I'm not sure we have any development in the future for doing that. It's a little more complicated, again, to concatenate sequences and batch them together. Of course, you can do it in sequence. It's not too cumbersome, but no, we looked into that and we just haven't developed that. So it's just one by one right now. So Jason wants to know, does the process redact sensitive species localities as designated by each Symbiota portal? No, it actually doesn't. If it's populated in those Darwin core fields, it's going to be pulling that data automatically from there. We are just actually pulling the data from the Symbiota tables, and that's why this online, or why this plugin module works outside of Symbiota. We're not really inside Symbiota code. We're outside it, which is a plugin, and we're just actually just sucking the data out. I could understand why you probably wouldn't want to do that, and there would be some mechanisms for people to just go into the SQN file, open it up in a text editor, and you could just remove that data manually and then resave the SQN file as a text file. I'm more than happy to show anyone how to do that. It's very, very easy to actually do. So you could redact that sensitive species locality post-file generation in your SQN file and then end up emailing that to GenBank. I don't think it's a good option to ask GenBank to remove it. It's just more work for them. But we could certainly submit them files that you don't want to show that locality data. And it's fairly easy to do that. Okay, so Hojun wants to know, let's see, you can insert source modifiers directly to FASTA file that can be acceptable for Sequent or Banquet, so maybe you can develop a tool to add the source modifiers to a FASTA file that is submitted to your portal. That would be a modified FASTA file that can be submitted through Banquet. Yeah, we're not really interested in doing that. Sorry. I've done that before in the past where I take source modifiers and I've added them to my FASTA file, and that just kind of defeats the whole purpose of this tool for automatically pulling the data and making it simple. Because what you're suggesting now, unfortunately, is retyping the data all over again, which you may introduce more errors into your FASTA file, into your source modifiers in your FASTA file, or you may accidentally put collected by in the metadata and collected by in the identified by field. And that's what we're trying to prevent by automatically pulling the metadata, is where you've already put the data in correctly in Symbiota. Why would you want to go retype it again and risk any errors? So no, we have thought about that, Hojun, but sorry, that's not going to happen. But very good questions so far. So it looks like we're getting on about 5 till 12 my time. We went on for about an hour so far. So I don't see anyone else typing, so this might be a good chance to wrap it up. And thank everybody again for joining the Symbiota Working Group webinar, and we look forward to seeing you at the next webinar. All right, Neil.
Yeah, and Andy.
Go ahead.
Oh, I just popped in the URL for next week's seminar, which will probably be a very different crowd of people, but it's Jonathan Amos is giving next week's one, and then in early April, Ann Barber will be giving one on the new data cleaning module that Ed has developed, and then we'll finish up with Kurt Dyerson I think in May, talking about Symbiota 2.
Awesome. That sounds great. I'll just remind everyone that this webinar will be posted at the iDigBio Symbiota Working Group website. And you can go there to listen to this again. Thanks. Bye.
Bye.